Hello everyone. Today we are going to talk about macromolecules. The term macro means large. So those molecules are pretty large, and we are going to talk about the main four macromolecules that we have within our body. The four main macromolecules that we have are carbohydrates, lipids, proteins, and nucleic acids. Let's talk about each one of them. We're going to start off with our carbohydrates. Carbohydrates are your carbs, and they are made out of sugars. Now, molecular formula-wise, what type of elements make up your carbs? You have mostly carbon, hydrogen, and oxygen. But basically, carbs are made out of sugars. We're going to talk about the different types of sugars that we have. Carbs are the main primary fuel for our body. Now, our body can actually use different macromolecules to break down and get energy, but carbohydrates are the primary because they are the easiest to break down. So, as I said, carbohydrates are made out of sugars. We have different types of sugars. One type is known as monosaccharides. Mono meaning one, saccharide meaning sugar. So those sugars are very simple type of sugars, and they're only made up of like one type of sugar. What are some examples of monosaccharides? Examples of monosaccharides are glucose, fructose, and galactose. Fructose is the simple sugar found in fruits. Now, notice something common between those three monosaccharides. You notice that molecular formula-wise, it's the same. All of these monosaccharides have 6 carbons, they have 12 hydrogens, and 6 oxygens. Now, does that make them identical? No. And actually, if you taste glucose versus fructose versus galactose, the sweetness level of each type of monosaccharide is actually going to vary. So which is more sweeter, glucose versus fructose, for example, fructose. So if you taste both of them, again, they're both going to taste sweet. At the end of the day, they're both sugars, but fructose is going to be much sweeter compared to glucose. And the reason why they're going to be different is because structurally they are structured differently. So even though they have 6 carbons, 12 hydrogens, and 6 oxygens, notice how each one of them is structured differently. And we always say structure determines function, and so different structure is going to allow them to have different functions. And in this case, again, if you taste it, they are going to taste a bit different. What do we call it when you have several molecules that have the same exact molecular formula, but yet they are a bit different? We call them isomers. So they have the exact same molecular formula, but structurally their arrangement of those elements are different. So you can see right here, those highlighted parts, those are the parts that are structured and arranged differently, for example, between glucose and fructose. Glucose is the primary fuel for our cells. When our cells use glucose, they get ATP, which is energy, out of it pretty fast. Now, we have to still watch out how much glucose we do consume, because if you consume glucose and then you go ahead and you break most of it down to get your energy, the person is not going to gain weight because you are breaking it down. But if you are consuming food that has lots of carbohydrates, and as we said, carbohydrates are made out of sugar, and let's say you don't really break it down, so your energy level is pretty low. So let's say you ate a pretty heavy meal of carbohydrates, and then let's say you just went to sleep or take a nap, your body is not going to break down as much of the carbohydrates that you've consumed because your energy now level is low. So Your body is not going to break down a whole lot of it. So what's going to happen is your body is going to start storing it. It's going to be stored as glycogen, and glycogen is mostly stored in your muscles and in your liver. And your body is also going to store it kind of like a long-term storage. It's going to store some of it as fat. And so, of course, if we eat a lot of carbohydrates, then again, we don't, let's say, energy level wise, it's not as great. Let's say someone is not really working out, and yet they are consuming too many carbs. They could start putting on weight because, again, the extra carbs are going to be basically stored as fat. Now, storing some of it in your muscles and in your liver, which is glycogen, is actually helpful because in case, let's say you are fasting and you haven't eaten anything, what's going to happen is your energy level is definitely going to drop. What is your body going to do to try to increase your energy level? Your body's going to go to your stored glycogen in your muscles and in your liver, and it's actually going to start breaking it down for you. If your body actually runs out of glycogen, then your body moves on to breaking down your fat. But the reason why your body is going to start breaking down glycogen first before it taps into your fat storage, because glycogen is going to be much easier to break down compared to fat. Fat is kind of really stubborn to break down. So let's say someone wants to lose weight and they cut off the carbs out of their diet, or they consume very little amounts of it. What's going to happen is the body is going to go to the stored glycogen, start breaking that first. Once it's done breaking down the glycogen, then it's going to move on to breaking down the fat. And actually, when you break down your fat, you are going to get a whole lot of energy. So monosaccharides, we talked about the three monosaccharides that we have, the glucose, the fructose, and the galactose. There is another type of sugar that we have, and it's known as disaccharides, di meaning two. We have sucrose, lactose, and maltose. Those disaccharides, they are basically made out of two monosaccharides bound together. Now, so you can see right here, to make the sugar known as lactose, the reaction that basically takes place is a glucose is going to bind to a galactose to make the lactose. Glucose is known as a monomer. Mono meaning one, so monomer meaning like one molecule. So if you get one monomer glucose and one monomer galactose, and what happens is a reaction known as the dehydration reaction. Dehydration is basically where water is going to be lost. When water is lost, that's going to help allow those two monomers to stick to one another. And what you create is something a bit larger, so we call it a polymer. Poly meaning many. So now you have a polymer, which is lactose. Of course, the polymers could vary in size. Like this is a very short polymer. You could have polymers that are made out of like 20 monomers bound to each other. Lactose is actually the sugar found in dairy products. So, for example, milk, and those individuals that are lactose intolerant, the problem is they don't have the correct enzyme to break down the lactose sugar. And so therefore what happens is the lactose starts to accumulate and it would start to ferment within the intestines, and that can cause the problems that lactose intolerant individuals would face. There are sugars known as polysaccharides, poly meaning many. We've got starch, cellulose, and glycogen. And so these polysaccharides, they're actually made out of lots of glucose monomers bound to one another or linked to one another. Of course, the starch, we kind of know starch. It's found in pasta, rice, potatoes, all of these would contain starch. And if you break down starch, so let's say you eat some potatoes, potatoes do contain starch. So when you consume those potatoes, your body's going to start chemically breaking down the starch in those potatoes. And when you break down the starch, what do you get? You basically get a bunch of glucose monomers. Cellulose is the polysaccharide found in plants. So plants do have cellulose, gives them a structure to their cells. When we eat plant products, we consume cellulose. But the thing is, we humans cannot break down the cellulose. But why is it important for us to eat cellulose? Because cellulose is basically going to act as our fiber. So when they tell you you got to consume fiber to have, let's say, easier bowel movements, the fiber is basically the cellulose that's found in those plant products. Even though we can't break it down, it's basically going to help push the contents within your GI tract again um, to help you, with the bowel movement. Glycogen, we talked about it. That's the stored sugar that we store it in our liver and in our muscles. And again, this is like a storage unit that we are going to have within our muscles and our liver in case your body needs some energy and you haven't been eating, your body can just tap into that storage and start breaking it down. And when you break down glycogen, again, you get a bunch of glucose monomers. Right here, you can see those polymers. We've got starch. So as you can see, it's a polymer because it's made out of many glucose monomers bound to one another. Glycogen is another polymer, and cellulose is definitely also a polymer. Now, take a look at them. Even though starch, glycogen, and cellulose, they're all made out of glucose, uh, structurally, they are different. And so they don't look the same, and that's why when your body comes to break it down, um, your body, for example, cannot break down cellulose, but your body can break down glycogen and starch. And the reason, again, your body cannot break down cellulose, somehow this structure right here, your body has a hard time breaking it down, and we don't have the correct enzymes to help us break it down. But again, as we said, we use it as fiber. So when we talked about making those polymers, for example, starch, we said that starch is made out of monomers linked together. Whenever a polymer is being made, the reaction that takes place is basically known as a dehydration reaction, where water is going to be lost. If you want to break down the polymer, the reaction is going to be reversed and the reaction is going to be called hydrolysis. Hydro meaning water, lysis meaning to break down. So to break down a polymer, all what needs to be done is water needs to be added to this reaction. So let's say that you've consumed some pasta. Pasta contains starch, and we know that starch is a polymer. Once you consume the starch, a hydrolysis reaction is going to start taking place and it's going to break down into its monomers, and in this case, it would be glucose. Let's say that you ate some meat. Meat contains a polymer known as protein. So your body is going to take that protein from the meat that you've consumed, and it's going to start breaking it down by adding water. And again, the reaction is called hydrolysis, and proteins are made out of amino acids. So those amino acids are going to be the monomers. Let's say your body wants to make a protein. What your body can do is take those amino acids and go through a dehydration reaction, lose water, and basically link those amino acids together to create a polymer, which is the protein. So as you can see, the reaction is reversible. Those monomers, if they are really like, small molecules, we call them monomers. If they are a bit larger, we can call them polymers. And you can have polymers binding to monomers to actually create a bigger polymer. Not all the carbs that we consume are digestible. We did talk about cellulose, and we said that our body cannot really break it down, but we do use it as fiber. Another carb that humans cannot digest is chitin, and chitin is, for example, found in crustaceans. So it's found in the outer part of the, like, let's say lobster or shrimps, etc. And again, we cannot break it down. Now, the second macromolecule that we are going to talk about are lipids. Lipids are basically fats. If you have right here in this flask, you have some oil and you have some vinegar, you will notice that the oil never mixes with the vinegar, so you see separation, and the oil is always going to float up at the top. That's because oil is hydrophobic. It does not dissolve and mix with water. But vinegar is hydrophilic. So if this right here, down here, you actually have some water and some vinegar, you're not going to see any separation because vinegar is hydrophilic. Hydro meaning water, philic meaning loving. So vinegar loves water, meaning that it can dissolve and mix in with the water. Lipids are known to be hydrophobic. They do not mix with water, and you will always see a separation between the lipids and anything that's hydrophilic. Now, when we consume fat, we can actually break down the fat that we consume, and we can actually get lots and lots of energy out of it. Lipids are not really as huge macromolecules as some of the other macromolecules that we've already talked about. And lipids, not all lipids are actually made out of the same repeating monomers. So for example, we talked about how carbs are made out of sugars. So if you take a look at starch versus glycogen versus cellulose, all of these polymers, they're actually made out of the same repeating monomer, which is glucose. On the other hand, lipids are not. So you can have different lipids, and each lipid is actually made up of a different monomer. And so let's go ahead and take a look at some of the different lipids that we have. One of the lipids that we have are triglycerides. Triglycerides are usually found in animal products, so cheese, meat, etc. would have triglycerides. Triglycerides are lipids that would make you gain weight if you consume too much of them. So if someone consumes too many of those triglycerides, they are going to start putting on weight. Not all lipids make you gain weight, by the way. So one of them that makes you gain weight is triglyceride. Now, why is it called tri? Tri meaning three. So you actually have three fatty acid tails, and it's bound to a glycerol molecule. Now, how would the glycerol molecule bind to the fatty acid? A dehydration reaction takes place where water gets lost, and those fatty acids are able to bind to the glycerol. When we talk about fats, we usually divide them into two groups, saturated versus unsaturated fats. Saturated fats are usually the fats found in animal products. So cheese, meat, etc., those would have saturated fats, and they tend to be solid at room temperature. Unsaturated fats, those are a bit healthier because your body has an easier time breaking down the unsaturated fats, and they tend to be in nuts, in fish, like the omega-3 in fish. You also have plant-based oils. Those are unsaturated fats. When you look at their structure, Both of them are triglycerides. So you have the fatty acid tails and it's bound to the glycerol molecule right here. But even though they're both triglycerides, structurally they're a bit different. Notice how saturated fats, their fatty acid tails are really straight versus the fatty acid tails of unsaturated fats. Um, they have like those kinks right here within them. That's because they have more double bonds. So when their carbons are bound to one another, so those black structures are representing carbons, and those sticking out, those are the hydrogens. Unsaturated fats have more double bonds compared to saturated fats. You can see right here, single bonds for the saturated fats. Having those double bonds causes their fatty acid tails to not look very straight, and actually we are going to, like when your body is consuming those double bonded triglycerides, it's much easier to break down those double bonds compared to breaking down those single bonds. And so therefore we say that consuming more unsaturated fats is healthier compared to consuming saturated fats. Of course, we still have to watch out how much unsaturated fats we eat, because again, too much of it and it will still make the person gain weight if they consume too much of it. There's another type of fat known as trans fat, or we can refer to it as hydrogenated oil. Trans fat is man-made. What they wanted to do with trans fat is they wanted to make a fat that would look like saturated fat, so it would look solid, like butter, but they wanted to make it have characteristics of unsaturated fats, which are the oils. So they wanted to make a fat that was kind of in between, like it's not very solid like butter, and at the same time it's not very fluidy like oil. They wanted to make something in between. You know those commercials that they say like, I can't believe this is not butter? Because they wanted to make it easier, let's say, to spread on your toast. So that, again, hydrogenated butter that they made, it looks like butter, but at the same time, it's much easier to spread on the toast. So how did they make that? They basically took the unsaturated fats, and what they did, remember how we said unsaturated fats have more double bonds? So you have more double bonds holding the carbons together. They wanted to break down some of those double bonds and turn them into single bonds to kind of make it more solid-like structure like the butter, like the saturated fat. So they wanted to break down a lot of those double bonds and turn them into single bonds. Well, how can they do that? They basically started to add hydrogens, and that's why the process is called hydrogenation. Hydrogenation because hydrogens are going to be bound to those carbons. And so they break down those double bonds, and instead of having double bonds, they will just have a single bond. But for the elements to be stable, they had to add hydrogens to still kind of keep keep all the elements stable. And when they did that, notice how unsaturated fats have like a lot of kinks because they have lots of those double bonds. So when they broke down those double bonds and they turned them into single bonds by the hydrogenation process, it's still not straight. So it's still not very straight like saturated fats. It does have some kinks in it because it's still going to have some of those double bonds, but it's kind of in between. So it's not really unsaturated and it's not really saturated. It's kind of something in between. And that trans fat is actually less healthier, way less healthier compared to saturated and unsaturated, because your body's going to have a hard time actually breaking it down. Your body has a hard time breaking down those fatty acids right there. And the reason why they use it a whole lot, especially in cookies and donuts. It turns out that using trans fat um, allows those products to have a longer shelf time. So therefore, you can see that box of cookies or that box of donuts on the shelf, and it says like you can consume it. Basically, it's not going to go bad for 2 years. And so trans fats definitely like elongates the shelf life of those products, and it does improve also the taste. So they found out like taste wise makes it taste better, but healthier, like health wise, it's not really healthy at all. Another type of fat that we actually have is cholesterol. Cholesterol is not a dry glyceride. So cholesterol is not made out of fatty acids. It's not made out of glycerol. It looks something like this. So as we said, lipids, they're not made out of the same monomers. So we talked about the triglyceride lipid. Now we're going to talk about cholesterol. And cholesterol is actually pretty important because it does help you make the sex hormones like testosterone and estrogen. Um, Now, we do have two types of cholesterol. So if you've done a blood test, you usually see the HDL and the LDL. And the LDL is the unhealthier one, and it can actually, if you have too much of it, it can start attaching to your blood vessel walls, and it would start narrowing your blood vessel walls, which is dangerous because if your blood vessels narrow too much, it can cause like a heart attack, it can cause strokes, etc. Um, and the cells in our liver, by the way, produce almost 90% of the cholesterol that we have in our body. Another type of lipid that we have, phospholipid. Phospholipids are not going to make you gain weight. Phospholipids are basically the major component of your cell membrane. The cell membrane is basically the boundary of your cell. It's that exterior portion of your cells. So again, having too many phospholipids do not make the person gain weight. It's basically going to help structure, like, the cell membrane of your cells. This is how it looks like. It's made out of two fatty acid tails bound to a phosphate group. A phosphate is basically a phosphorus element bound to oxygens. So these right here are oxygens. And so usually that phosphate head is hydrophilic. It's able to withstand water and mix well with water. The fatty acid tails, those are the hydrophobic tails because they do not like water. The third macromolecule that we are going to talk about is protein. We have so many different proteins throughout our body, and those proteins have a whole lot of different functions. Some of the proteins that we have give us structure, like they help give structure to your cells. An example would be those proteins in our cells known as the microtubules and the microfilaments. Those definitely give structure to your cells. We have proteins in our hair and nail known as the keratin protein. We also have proteins that help protect us. An example would be a protein that we have in our blood known as the defensin protein, and they basically help us fight off infections and any microorganisms that might have invaded our body. We have some proteins that can help you coagulate your blood. So if you've injured yourself, those proteins would help coagulate your blood so that you could stop your bleeding and help you heal. We have some proteins that would help us regulate things in our body. So a lot of the hormones that we have in our body, they are actually proteins. We have proteins that would help us with contracting our muscles, such as the actin and myosin proteins. Um, we have some proteins that would help us transport things in our body, such as your hemoglobin. The hemoglobin protein helps transport oxygen throughout our body. So as you can see, the we have so many different proteins spread throughout our whole entire body, and the functions of those proteins are immense. Now, what are proteins made out of? What are the monomers of proteins? Well, the monomers of proteins are amino acids. We actually have about 20 different types of amino acids. Now, does that mean we only have 20 proteins? Absolutely not. It's kind of like the alphabets. We only have 26 letters in our alphabets, but we are able to say so many different words. That's because it depends how many letters you choose and how you are going to arrange them. You can, again, come up with an immense amount of words. Same thing with proteins. Even though we only have 20 amino acids, depending on which amino acid is going to be linked, and depending on the arrangement and the order of these amino acids, you are going to be able to make so many different types of proteins. This is how an amino acid looks like. So an amino acid has this carbon right here bound to a hydrogen. We usually refer to it as the central carbon because it's in the center. It's bound to an amino group, which is a nitrogen bound to hydrogens. And then you have a carboxyl group, so a carbon bound to a couple of oxygens. And then there is this part right here of the amino acid known as the side chain. Now, as I said, we have 20 amino acids. All 20 of them have the following. All 20 have an amino group, a central carbon, and a carboxyl group. What's different about those 20 amino acids are the side chains. Those are some examples of side chains. And having different side chains is basically going to give different characteristics to each amino acid. So if the side chain looks like this, then the amino acid is called alanine. If this is the side chain, then this amino acid is called glycine. And again, this huge side chain right here, this is tryptophan. Tryptophan amino acids are usually found in turkey meat, and when you break it down, that's actually what makes you feel sleepy after eating turkey. Now, when you have so many amino acids bound together, we call that chain of amino acids bound together, we call it a polypeptide chain. Poly meaning many, peptide are basically the bonds that are going to hold the amino acids together. So if this is an amino acid right here, you have another one right there, what reaction is going to take place to bind those two amino acids? A dehydration reaction, where water is going to be lost and those amino acids are going to be linked together. Now, the bond that's going to hold these amino acids together is known as a peptide bond. So when you have a protein, which is going to be made out of so many amino acids linked together, you end up with so many peptide bonds. So we call the chain a polypeptide chain. Now, for protein to basically be functional, the protein has to go through many structural steps to be basically become functional. And so the very first step is the primary structure. The primary structure of a protein is basically a polypeptide chain. It's a chain of amino acids bound together via those peptide bonds. Now, this primary structure is really important because the correct amino acids, first of all, must be chosen. And then another thing is that those amino acids must be in the correct sequence. So arrangement-wise and sequence-wise, it has to be correct. If it's not, disorders can actually start happening. An analogy, just to kind of give you an example of, a polypeptide chain, let's say you want to spell out the word protein. Now, for the word to, for you to basically spell out protein, you need to choose the correct letters. That's one thing. And then another thing is after you've chosen the correct letters, you must make sure that they are in the correct order. If you forget the letter protein, it's not going to spell protein anymore. If you switch out the letter O with the letter T, you put them in the incorrect order, again, it's not going to spell out the word protein. And this can actually happen inside of our body when your body is making those proteins. So that would be a mutation, and those mutations could actually be heritable, like you've inherited that mutation, and so whenever you make that protein, this protein is not going to function properly because there's something wrong with the sequence of the amino acids. An example is individuals that have sickle cell anemia. Individuals that have this disorder, basically their hemoglobin protein doesn't function properly in transporting oxygen. And the reason why their hemoglobin is not functioning properly, because the hemoglobin protein, it's a protein, so it's made out of amino acids. These individuals, one of their amino acids, known as glutamic acid, gets switched to valine. So instead of having glutamic acid, let's say right here, instead they have valine. Even though it's just one amino acid, that's really, that's incorrect. One amino acid that's incorrect is going to lead to a problem, and the whole entire protein doesn't function properly. Now, after the primary structure, that polypeptide chain is going to start having a shape, and we call it a secondary structure. So the secondary structure is the polypeptide chain is going to start twisting itself, or it's going to start kind of folding itself right there. So we either call it a helical structure, like when it's twisted like this, or we call it beta pleated sheets, when it basically looks like folded sheets right there. Again, something could go wrong on that step, and the polypeptide chain might not start twisting properly, or it's not folded properly, and that can cause the protein to not function properly. Then the secondary structure is going to fold itself even more, and it's going to start kind of getting a complex three-dimensional shape. Now, so far the protein is not functional yet. So again, for protein to become functional, you first have to have the proper primary structure, and then the correct secondary structure, and then you move on to the third structure. Again, the protein is not functional yet. After it kind of gets its three-dimensional shape, we move on to the quaternary structure, where you can have more than one polypeptide chain bound together. After that quaternary structure, like once the quaternary structure is ready, then then basically the protein is now functional and ready to go ahead and start its job. Whether its job is to help you transport oxygen, whether its job is to give structure to your cells, again, depending on which protein is being made, it's definitely going to have a different function. Something could go wrong with any of the steps that we've been talking about. So one thing that can go wrong with the quaternary structure formation is one of the polypeptide chains might be missing. So another example about the hemoglobin protein is actually made out of 4 polypeptide chains linked together. Now, not all proteins are made out of 4 linked polypeptides. Again, each protein is different, but hemoglobin needs to have 4 polypeptides. This order can happen where basically one of the polypeptide chains is missing. So instead of having 4, those individuals would have 3 polypeptide chains linked together, and again, that's going to cause their hemoglobin not to function properly when it comes time to transport oxygen throughout their body. The last macromolecule that we are going to talk about are nucleic acids. An example of nucleic acids that we have in our body is basically DNA and RNA. Now, when it comes to speaking of the monomers of those nucleic acids, the monomers are known as nucleotides. So a nucleotide is the monomer of our DNA and our RNA. What is a nucleotide made out of? A nucleotide is made out of the following. It's made out of a sugar molecule. Now, the sugar molecule is going to be different if we're talking about DNA versus RNA. The sugar in our DNA is known as deoxyribose. The sugar in our RNA is known as ribose. So DNA and RNA, they both have sugars. It's just that the sugar is different. This sugar molecule is bound to a phosphate group. Both DNA and RNA have a phosphate group. Nothing is different about the phosphate group for the two. And then the sugar molecule is also bound to a base. So the four bases that we have for our DNA are the following: adenine, thymine, guanine, and cytosine. Only one base is going to be bound to one sugar at a time. So right here, if we have a long strain of our DNA, this is how it's going to look like. So we have a backbone for our DNA, and the backbone for our DNA is basically sugars and phosphates. That would be the backbone right here. And then sticking out from each sugar molecule, you're going to have a base. And again, the bases for our DNA are the following right there. Our DNA is a double helix, meaning that we have two strands of DNA that are linked to one another and they start twisting. So let's take a look. So right here, you can see that our DNA is a double helix. It's actually made up of two DNA strands twisted and linked to one another. This basically represents the backbone of our DNA, and the backbone of our DNA, it's made out of the phosphates and the sugar groups. What's holding those backbones together are basically the bases, the nucleic bases. So adenine always binds to thymine and guanine always binds to cytosine. So if this is one of your DNA strands and you have A on this side, you definitely have to have a thymine on the other side. So, and that's how, again, they are going to be linked together. Those bonds that are holding them together are the hydrogen bonds. That's just another image showing you the DNA, which is a double helix, and you can see the backbone of the DNA made out of the phosphate groups bound to the sugar groups. And then you can see the nucleic bases linked to one another, holding those two strands together. So adenine always binds to thymine, and cytosine always binds to guanine. And that's why at the end, we say that humans' DNA, it's a double helix, because it's two strands, and then helix because it starts to twist, as you can see. So we talked already about our DNA, and DNA again stands for deoxyribonucleic acid. Deoxyribose, that's basically the sugar. There is another type of nucleic acid we have, and it's our ribonucleic acid. The monomers of ribonucleic acid is also known as a nucleotide. The nucleotide for the RNA is going to have a sugar group. That sugar group, instead of calling it deoxyribose, we call it ribose. And it also has a phosphate. The phosphate group is the same for both DNA and RNA, and then it has the bases. Now, RNA has one different base compared to DNA. RNA has adenine, it has guanine, it has cytosine, but it doesn't have thymine. Instead, it has uracil. So right here, you can see some structural differences between DNA and RNA. The first is DNA is a double helix. RNA is a single-stranded molecule, so only one strand. Another structural difference between DNA and RNA is that the sugar of DNA is known as deoxyribose sugar. RNA, its sugar is known as ribose. Another structural difference is the bases. DNA has thymine. RNA doesn't have thymine. Instead, it has uracil. DNA is what holds our genetic information. So all of your genetic information is basically stored within your DNA. Why is RNA important? To make proteins, we actually have to go through a process known as transcription, where we take the information from our DNA and we transcribe it and we make an RNA out of it. Once we make RNA, another process known as translation takes place, where you translate your RNA into a protein. So you take the information, you basically translate the information within your RNA and you translate it into a protein. So without RNA, we can't make proteins. And as we said, proteins are extremely important. We can't survive without our proteins. So both DNA and RNA are really, really important. But even though they're both monomer-wise, they're both made out of nucleotides, there are some structural differences between the two. So some structural differences, as we said, the sugar molecule is different between DNA and RNA. The structure, like DNA is a double helix, RNA is just single-stranded. And then, of course, the nucleic acids. DNA has thymine, RNA has uracil instead.